Welcome back to the vaccine design course. Our next step is the prediction of antibody protein. So here what we are going to do is antibody modeling or to find the antibody protein. Now antibodies, they are Y-shaped proteins which are essential for immune response. Their capability to recognize antigens with high specificity makes them excellent therapeutic targets. So understanding the structural basis of antibody-antigen interaction is therefore crucial for improving our ability to design efficient biological drugs. So let's first go to rcsb.org and search for the antibody protein. Okay, why we are doing it is for the antibody modeling. So let's start with it. So I've searched for RCSB, go to its homepage, and what we are going to search here is we are searching for antibody. Okay, search for antibody. So for antibody modeling, what we need is the FASTA sequence, right? So we need the FASTA sequence. Let me tell you that there is no server available where we can manually design our own antibody. So that's why we are taking from the database. Okay, so now we have searched for antibody. Now click one antibody from the results. Okay. Okay, so let's click the first antibody. Okay, now when you click for the antibody, you can see the antibody information. So here what we need is the organism as Homo sapiens, which we already have. Okay, the organism, it says Mas musculus and Homo sapiens. So Homo sapiens is involved. Now to take its FASTA sequence, click on display files, then FASTA sequence. Okay, so here is our antibodies FASTA sequence. So now you have chain A and chain B. Anyway, control A, control C, which means we are copying the FASTA sequence, and let's paste it in the notepad. Okay, so let's paste it in the same notepad. One second. Okay, let me just do it again. Okay, yeah, so let's click undo and see. Okay, anyway, you have your alignment which is saved in a different folder, right? Okay, so let it be like that. Or let's see, let's again do the undo, and yeah, and then paste your sequence information. Okay, so which is chain A and chain B, right? So now you have your antibody protein sequence. Okay, so let's name it as antibody protein 1. Okay, you can either save it in different notepad and save it. Okay, so now what you need to notice, you already have a 3D structure in RCSB, but here what we are doing is we are learning how to design an antibody. Normally what you can do is you can take the 3D structure that is available in PDB. You have already designed antibody and dock it with your antigen, but we need to learn, right? So yeah, so now this was the step that we did. Now to design the antibody, we are now gonna go to another server named SABPRED. Okay, either you can go to this particular link or search for SABPRED now. So Google SABPRED and take the first result. Now what you need to do is you need to paste the heavy as well as light chain, right? You have the chain A which says L, which is the light chain, and you have the chain B which says H, which is the heavy chain, right? You need to paste it. So this is the home page of SABPRED. So go for FE modeling A body builder. Okay, this option. Okay, so this was the link that I've given you. If you click this link, you'll be directed to this particular page. Okay, now here you can see the sequence submission form where you need to paste your heavy chain sequence. So now let's paste the heavy chain sequence, this, right? Okay, so this is your heavy chain sequence. So paste your heavy chain sequence here, and let's see when you click on load example, it says that there is no need for FASTA header. So without the FASTA header, you need to paste the heavy sequence here. So remove the FASTA header. Okay, so remove the FASTA header, and now for the light chain, again take your light chain, which is just one single line, which is the light chain, and then paste it under the light chain sequence. So now you have pasted both the heavy as well as the light chain. Then if you want, you can give your job name, and then a method. It's already, you can click the question mark symbol to know about this, and then after that click on model. So here the results are ready. Now you can see what the bodybuilder used, that is the software, and now you can see under the results, you can see your model and various annotations. So the rank 1 model, that's your antibody protein in PDB format. You can download this thing by clicking the download button. So click the download button wherein you can download your PDB, show in folder, and then you rename it as your antibody model, okay, or you can rename it as SABPRED, because we are using another server also, SABPRED antibody, and save it in your file. So I've saved it as SABPRED antibody. Now other interpretation of the result is you have your template details, then the sequence alignment which is done, and all other things, okay. But what we needed was our antibody protein structure, right? Okay, so that's about the SABPRED server, right? Next, another server that we'll be looking for to see the antibody model or to get the antibody model is Lyra IEDB. Okay, go to this particular link or search for Lyra IEDB. So search for Lyra IEDB, go to the homepage for Lyra, which is lymphocyte receptor automated modeling. Okay, that is the full form of Lyra. Now paste the first as well as the second chain sequence here. Okay, so let's paste the first sequence, which was chain A, right? So paste the first sequence here, and again paste the second sequence. Paste the second sequence in the second chain sequence, or you can choose file as well and then click the submit button. Here in the results, you can see the input sequence and then the summary of it, and then the heavy chain as well as the light chain alignment, and the 3D structure view also. It's been loading. Let's wait for it to get loaded. Yeah, so you can see the 3D view. You can download this PDB model file. So click the download button and then show it in the folder, and then rename it as Lyra antibody, and then save it in your folder. Okay, so your SABPRED antibody as well as Lyra antibody is here in your folder. So coming back to our slide, so this is how you get your antibody structure. Now let's take, I told you, right, you already have your antibody, so let's take the already available antibody structure which is available in RCSB. Okay, so go to RCSB, search for RCSB, which is again here. Okay, or go to this particular link that we have already found out. Okay, so it's 1FL5 that we have selected, right? So you have download files here, and then you have PDB format which is written. So click on PDB format, then you will get your file. Show it in folder. So this is antibody in RCSB, which is already there. So save it like that, and then or you can say RCSB antibody. Okay, save it and then paste it in your folder. So yeah, so now you have done till this step, right? So now you know how to take the antibody structure, how to download it from 2 servers, and then now, but you already know how to take your or retrieve your antibody protein model which is already there, right? So now the next thing is you have your antibody or you have your antibody protein prediction. Now is to do the functional analysis of the antibody protein model. So for that you need to check the stability, like how we checked the stability of the antigen protein model, you have to check the stability of the antibody protein. So an antibody's stability, it greatly influences its performance, right, its specificity and affinity. So that's why it's really important to check the stability of the antibody protein model. To check the stability, again we need to go to mol probity and then upload the PDB file. Okay, so again search for mol probity and then take the first result, and then you need to upload the PDB file either from Sapred or Lyra or the protein model which we already have. Okay, so let's take the Sapred model, or let's just take the antibody model which we already have, RCSB. Okay, RCSB, and then you need to click the upload button. Okay, click upload. By the time you do the exercise by taking out the Sapred and Lyra antibody. Okay, and then press the continue button which we have already did in your antigen protein body stability checker. Here again press the analyze geometry, and then if you want hydrogen van der Waals contact, you can click that, and then press on run programs to perform these analysis. Click OK button, and then look for the Ramachandran plot values. So like we already mentioned, if we met the goal, then it means that the antibody model is stable. Okay, now see the Ramachandran favored. As you can see here, the antibody protein structure is not that great because it's not meeting the goal, right? When we look at the Ramachandran outliers as well as Ramachandran favored, see that if it has green color, if it's green color, then it means it has met the goal. But yeah, so this is how you need to look for the stability of antigen as well as antibody protein. Okay, so that's all about the antibody structure prediction as well as functional analysis of antibody protein model. Thank you.